Hi, good morning, everybody. It's Ms. Corcoran, and we are going to finish Chapter 8, the photosynthesis chapter in your book today. Last time, in the last video lesson, we learned about the light reactions and the process of oxidation and reduction. And when something gets oxidized, it loses an electron, but that electron doesn't just float around in space, hanging out, doing nothing. That electron has to land somewhere, and wherever it lands, that particular thing has been reduced. So oxidation and reduction always go together. So we drew in the drawing photosynthesis lesson, and hopefully you can draw this forwards and backwards and in your sleep. This is the chloroplast. The light reactions take place in the thylakoids. The reactant of water is used here. We know it's split. It leaves as oxygen gas. And then we make these two intermediate cellular energy and electron carrying molecules of ATP and NADPH. And the driving force of all of that is light energy. Basically, the light reactions convert light energy into these two intermediate cellular energy products, and in the byproduct, create oxygen. So also review, this is the thylakoid diagram up close. I cannot stress to you enough the fact that you absolutely unequivocally must know every membrane protein in here. It kind of sounds like a lot, but it really isn't. You need to know PS2. You don't need to know these individually by names. You just need to know that it's the ETC, also called the cytochrome complex. You need to know this is PS1, and that over here, that's like a really short electron transport chain. We don't really even talk about it. We just kind of say up and over to NADP plus reductase, which helps reduce NADP plus into NADPH. You need to be able to tell the story of the electrons, that, excuse me, photons of light excite the electrons in the chlorophyll and they jump up. Well, essentially that chlorophyll has been oxidized. That was oxidized, this electron receptor has been reduced, and then that gets oxidized, this gets reduced, that gets oxidized, that gets reduced, that gets oxidized, that gets reduced, that gets oxidized, and that gets reduced, because this has also been oxidized by light at the same time. So when this first photosystem 2 chlorophyll has been oxidized, we need to replace it because it doesn't like having that missing electron. So the process of photolysis actually oxidizes water, and we take the electron from water, we add those protons to the thylakoid space, and we release oxygen gas. In the meantime, when those electrons go wee down the ETC, they are driving the active transport of protons, more and more protons into the thylakoid space until we get a great big pillow in the thylakoid. And at the end of the electron journey, after PS1, it jumps up and over to NADP reductase, and we reduce NADP plus into NADPH. That is one intermediate product from the light reactions. Meanwhile, we've packed this thylakoid space full of protons, and they all want to get away from each other because they're packed in there. And the only exit is through ATP synthase. It's easy for them to leave because everything in the universe wants to go from high to low. There's a high concentration in here, and they go wee down ATP synthase. Literally, it turns a knob that's going to phosphorylate ADP with a phosphate and create ATP. So voila. We have our two intermediate products that are going to go to the Calvin cycle. So what is the Calvin cycle? The Calvin cycle is actually the anabolic, the building process of photosynthesis. It's the actual building of the sugar. It builds sugar from smaller molecules using the energy provided by ATP and the reducing power of those electrons from our NADPH. And it also takes some hydrogens from the NADPH as well. So that's how we build the sugar. What do you specifically need to know about the Calvin cycle? First of all, you need to know that the three parts are carbon fixation, reduction, and regeneration. You need to know there are three parts. That the end product, technically speaking, of the Calvin cycle is a 3-carbon sugar precursor called glyceraldehyde 3-phosphate, or G3P. That's not glucose, is it? How many carbons are in glucose? 6, C6H12O6. Glyceraldehyde 3-phosphate is a 3-carbon compound. So naturally, it takes 2 of these G3Ps to make 1 molecule of glucose. Now, those of you that love numbers and love math are not going to have a problem with the carbons in all of the Calvin cycles that happen in the stroma. Those of us who are challenged with numbers and math, your head's going to want to explode. Let it go. Trust me, if there are C6H12O6, if there are 6 carbons in glucose, how many molecules of carbon dioxide do we need to make 1 molecule of glucose? You need 6. Okay, 1 carbon dioxide has 1 carbon. Glucose has C6, 6 carbons. You need 6 carbon dioxides to make 1 glucose. There's a lot of other stuff in there. Don't worry about it, okay? Just know that you need 6 carbon dioxides to eventually make 1 glucose. All right, so generally speaking, and that's what we put here, you need 6 molecules of carbon dioxide in order to make 1 glucose. So basically, the cycle has to turn 6 times. But remember, there's not just one Calvin cycle. We show you in our pictures, sorry, 1 Calvin cycle. We also show you 1 stack of thylakoids. There are a whole bunch of thylakoid stacks all throughout the chloroplast, and all of the space, that gelatinous stroma space all around the thylakoids, there are Calvin cycles happening right and left, so many of them. We just represent it as one little wheel cycle diagram. So just let it go, let the math go, and trust me. If you need to make 1 molecule that has 6 carbons, then you need 6 carbon dioxides to do it, because each carbon dioxide has 1 carbon. So hopefully I have warned you against getting a headache about the math stuff. So here is that we're done with the light reactions. We have made ATP and NADPH, and we're sending it over. I kind of consider this, we're paying the workers now. We had to do the money, what do you call it when you, oh gosh, my gosh, the word has left me. When you are switching money. I don't know, you know it. It's coming out of you, but it's stuck in my head. So we've done the money conversion of light energy into cellular energy, and now we can actually pay the workers to build the sugar. So there are 3 parts, like I said, and I'm going to go through it just with these words, and then we're going to draw it together. So get your piece of paper ready, because you're going to draw what I draw in a minute. So the first part is carbon fixation. When we fix carbon, so where is carbon right now? Carbon dioxide. It's all around us. Okay, it's in the atmosphere. Carbon dioxide is everywhere. So it's no big deal to grab it from the atmosphere and attach it to a pre-existing carbon-based molecule already in the plant cell. So that's what carbon fixation is. We're actually, if here is my pre-existing carbon-based molecule, and this is an enzyme that's going to attach the two. Carbon is in the air, and I got one, and I just attached it to a pre-existing molecule. Okay, the molecule that pre-exists is called ribulose bisphosphate. Nobody really says that. That's too much. We call it RUBP. So we take the carbon dioxide that's in the air. I'm an enzyme called Rubisco. Okay, I'm going to facilitate this, and this is my RUBP. Carbon dioxide's in the air. Rubisco goes, gotcha, and we're attaching it to RUBP. Perfect. We have just fixed a carbon dioxide from the atmosphere into an organic molecule within the plant. Done. Step 1, done. So here's how it looks. Don't freak out about the names of the molecules throughout the Calvin cycle. I'm going to let you know the molecules you need to know. So phase 1, carbon fixation. You need to know Rubisco. That's the enzyme that grabs it. You need to know RUBP. Just for fun, look at how many carbons are on RUBP. 1, 2, 3, 4, 5. There's 1 carbon in carbon dioxide. This shows 3 carbon dioxides entering. Remember, there are plenty of Calvin cycles happening all over the place, but we're just going to talk about 1 for now. When we grab 1, we're going to make a 6 carbon molecule, 1, 2, 3, 4, 5, 6, that immediately splits into 2 3 carbon molecules. You don't need to know the intermediate names, trust me. If you are a math person, I'm just going to kind of speak your language for a minute and help you account for that. Okay, so fixation. We literally grabbed it, Rubisco, the enzyme, grabs carbon and attaches it to RUBP, ribulose bisphosphate. That's exactly what's happened here. There's my hand enzyme grabbing carbon dioxide in the air, attaching it to RUBP, and then we have a 6 carbon molecule that's kind of unstable, then immediately splits. We don't really care about that. I mean, it's good to know, but we don't need to know it. We don't need to get bogged down in those details. The next part is reduction. Remember oxidation, reduction? So if we're going to reduce a molecule, what must we do to something else? We must oxidize it. Well, guess what? We've got NADPH, which I kind of picture as a waiter or a waitress with a tray. And on the tray, there are electrons and hydrogens. And they're like, here you go. Would you like these electrons? You may have them. And when you take the electrons from me, you are reduced, but I am oxidized. So this is where our NADPH and some of our ATP comes in. This is reduction. Once again, we're not worried about all these intermediate names at all. You just need to know that what's going on right here is phase 2, and it's reduction. We are throwing electrons and hydrogens at this molecule. At the end of this phase, so after fixation and reduction, we've actually made our 3-carbon sugar molecule, G3P. Remember, it takes 2 G3Ps to make 1 glucose. No big deal, because each G3P only has 1, 2, 3 carbons. Okay, if we stopped here, it'd be game over, baby. Photosynthesis would end. Because what did we have to start with? So, ta-da, yeah, we've made the sugar. That's the whole purpose of photosynthesis. But if we don't go any further, we're never going to be able to do it again because we had to start with RUBP. Well, we took the RUBP and we turned it in to glyceraldehyde 3-phosphate. We've got to remake RUBP. This is where the math part gets confusing because we've just spit out 2 3-carbon molecules and we have to somehow generate 5 more carbons in our RUBPH. Or RUBP. This is where you have to remember that the stroma has lots and lots and lots and lots of Calvin cycles going on. And so all you need to know is that if glucose has 6 carbons, then we have to fix 6 molecules of carbon dioxide ultimately to make 1 glucose. Okay, you don't need to get all caught up in all the intermediates. You need to know that it gets spits out as G3P. So how do we regenerate RUBP? Through a process called regeneration. And this is the 3rd part of the Calvin cycle. So we rearrange some molecules of G3P and we regenerate our initial carbon dioxide receptor or acceptor of RUBP. This requires ATP. So luckily, we made some ATP in the light reactions and we can power that. So once we have made some G3P, that's going to be combined with other G3P from other Calvin cycles that are all going on simultaneously in the stroma. And we're going to have enough carbons in the stroma to regenerate that RUBP. Because if we don't have RUBP, then we can't grab carbon dioxide from the atmosphere and fix it in the initial steps of carbon fixation with the help of Rubisco. So I believe that's it for the Calvin cycle. All right, I'm going to switch over here to the document camera. Okay, that's your overall of the Calvin cycle. And this is what you need to be able to draw. Let me see if I'm centered. Okay, we have CO2 coming in. We have RUBP pre-existing. Okay, that's our ribulose bisphosphate. We need to join, and that's 5 carbons. That's obviously 1 carbon. We need to join RUBP plus carbon dioxide. Who does that? Who helps us? An enzyme called Rubisco. If I have underlined it in green, you need to know it. When this happens, when we join the carbon dioxide to RUBP, that's called carbon fixation. Okay, we have fixed a molecule of atmospheric carbon dioxide and we have attached it to a pre-existing organic molecule in the plant. That's carbon fixation. Then we are going to throw some NADPH at it, and we're going to throw some ATP at it, okay, in the process called reduction. I guess I should underline that in green. So that would be step 1. That's step 2. Notice how I didn't include any of the intermediates right here. We don't need to know it. You need to know big picture of fixation, reduction, generally what's happening there, and then regeneration. Remember, at this point, we are spitting out a 3-carbon molecule of, you can call it G3P, or its real name is glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate. It's 3 carbons. We need 2 of these to make 1 glucose. Well, don't worry about it because we've got Calvin cycles going on all over the stroma. So easy peasy, we can recombine with other G3Ps and make our glucose. No big deal. But more importantly, if we want to do this again and again and again, and that's the nature of a cycle, is that we have to regenerate our RUBP in the process of regeneration. And we're not even worried about the fact that it takes ATP to do that. Okay, so our 3 steps. Fixation, where we grab carbon dioxide. Rubisco is the enzyme that helps attach it to RUBP, and boom, we have fixed atmospheric carbon dioxide. Then we're going to throw some electrons and hydrogen at it and use some ATP, and we're going to spit out a sugar precursor called glyceraldehyde 3-phosphate. Okay? It's only got 3 carbons, so we need 2 of those to ultimately make 1 glucose, but it's no big deal because there are Calvin cycles going on all over the stroma. Lastly, we must regenerate the RUBP so that we can grab another molecule of carbon dioxide and keep making the sugars that build the plant cell walls, that power the plants, and ultimately power us. Okay, so that is it for the Calvin cycle. Let's talk about a couple of adaptations in areas that are really hot. I think if you recall your transpiration lab, there were some conditions in which the stomata of a plant, actually that's the opening where the gas exchange takes place. So carbon dioxide comes in, oxygen leaves from that point, where the stomata actually closes in an effort to prevent water loss. Because in really hot areas, gas is not only being exchanged in the stomata, but evaporative water loss via transpiration is also occurring through the stomata. So the closing of the stomata is a good thing to actually help preserve water from being lost, but it's a bad thing because it reduces the amount of CO2 for photosynthesis and it causes oxygen to build up when photosynthesis does happen. And in situations like that, there's a process called photorespiration that occurs. And basically it's just when Rubisco gets confused. Okay, and it is a wasteful, ATP wasteful, energetically wasteful process. Excuse me. Okay, so let me just give you a couple of terms. C3 plants are normal plants, okay? These are not plants that live in really hot areas. These are not plants that experience enough photorespiration to have evolved something different. They still experience it, and the plant just kind of chalks it up to, well, I just got to use some ATP and undo what I've done. But C3 plants are named because when the initial fixation of carbon occurs, it forms a 3-carbon compound. Okay. Well, photorespiration is when Rubisco, remember Rubisco is the enzyme that helps to grab the carbon and fix it onto RUBP. It gets confused and it grabs oxygen instead. So it fixes oxygen and it makes this compound that the cell needs to spend ATP on to undo it. That's a waste. So it consumes ATP. How is this possible? That seems kind of weird. Well, if you think about it, Rubisco evolved. It came on the scene essentially when the atmosphere didn't have much oxygen because the oxygen came from photosynthesis. And Rubisco is important in driving photosynthesis. So Rubisco didn't really have to be very choosy. There wasn't a ton of oxygen in the atmosphere when plants first evolved the Calvin cycle. So Rubisco didn't have to think about it, and Rubisco can easily get confused. So it's an evolutionary relic that some plants just have to deal with. But in areas where the plants have a ton of photorespiration, they would be spending so much energetically trying to undo the photorespiration that plants that evolved a different strategy have been more successful. So C4 plants actually separate carbon fixation from the rest of the Calvin cycle by separating them into different cells. And they're called C4 plants because when carbon is initially fixed, the first product is a 4-carbon molecule. Remember, C3 plants make a 3-carbon molecule. So that's just where the name comes from. An enzyme called PEP carboxylase has a very, very, very high affinity for carbon dioxide. It does not get confused. It took a while for plants to figure out that PEP carboxylase worked better than Rubisco. And by then, as Crash Course says, plants had gone all in on the Rubisco, so only a few plants actually evolved PEP carboxylase. So it has a higher affinity for carbon dioxide. It binds to carbon dioxide and creates a 4-carbon molecule called oxaloacetate. That is a mouthful. You will see that molecule again, actually, when we talk about respiration. And I think it's super cool that we see molecules created in the chloroplast that we see created again in the mitochondria. Amazing. So basically, C4 plants fix sugar fix carbon and create the sugar spatially separated. They keep them separated into 2 different cells. Another type of adaptation are CAM plants, and it's called, I can't even say it, crustacean acid metabolism. Nobody says that. We call them CAM plants. CAM plants actually prevent water loss by closing their stomata during the day because they're in such hot climates, and they open it at night. So no big deal, easy peasy. We can fix carbon all night long. Well, remember, ladies and gentlemen, that yes, we can fix carbon at night, but we can't make the sugar until what? Until we've got, I'm trying to do this backwards, until we have our ATP and our NADPH, which come from what? The light reactions. So technically we do need light to make sugar. So what they do is they open their stomata at night and they incorporate carbon dioxide into an organic acid. And then during the day, when their stomata are closed, they undo that incorporation, and that carbon is then used in the Calvin cycle. So they actually temporally, or by time, they separate fixation and sugar production. You know, I like a picture way better than anything else. So here you go. Sugarcane and pineapple. Sugarcane grows in the tropics. Pineapples grow in the tropics. Those are both very hot climates where photorespiration was really an issue, such that plants that evolved these adaptations have done better. So in C4 plants right here, we've got spatial separation of carbon fixation and the Calvin cycle. Okay, no big deal. In CAM plants, we have temporal separation of carbon fixation at night. And then during the daytime, when we can make ATP and NADPH in the light reactions, that's when we can power the Calvin cycle. Okay, so why do we care about photosynthesis? Well, obviously it powers the entire Earth and most ecosystems except the ones at the very deep sea bottom of the ocean that are powered by the heat of the Earth. Sugar is made in the chloroplast. It supplies chemical energy and carbon skeletons to synthesize organic molecules. Plants are made out of products that they create from taking carbon in. Plants are literally made from the carbon in the air. Think of the biggest sequoia tree, a picture of the most gigantic tree you've ever seen. The carbon in that plant came from the air. The carbon that makes up the mass of that plant came from that initial fixation into the organic molecule during the Calvin cycle. In addition to food, it produces oxygen. So we really do care quite a lot about photosynthesis. Here is an overview, again, light reactions, the most useful, useless byproduct ever, oxygen. It sends these intermediates over to the Calvin cycle that power the reduction and synthesis of sugars, and of course, the regeneration of our carbon acceptor, RUBP. And after those molecules are spent, they are sent back over to the light reactions where they can be powered up again, where we can rephosphorylate ADP and we can re-reduce NADP plus to create these molecules that need to go back to the Calvin cycle. So even though these are sometimes called the dark reactions, this cannot run without these intermediate products from the light reactions. Once again, you need to be able to draw this picture with your reactants up top and your products down low and your intermediate products here and here. This is the basic overview. You need to be able to understand what happens in the light reactions and the Calvin cycle. You need to be able to draw, and I don't have another picture of it, that thylakoid membrane. And I'm posting on Schoology a blank thylakoid membrane for you to label and draw the process. It really is quite easy if you think about it as a story. We've all heard stories from our friends, and we can repeat those stories when we want to tell someone else. So if you think about it as the story of the electrons and the story of the hydrogens, it's a lot easier to digest than, oh my gosh, it's a really complicated biochemical process. These last two pictures are just connections to a working cell. We see active transport using ATP across the cell membrane. We see passive transport of the gases in and out, and we see some of the gases being used in these organelles. Lastly, the first discussion on the light reactions, we talked about linear electron flow. This is what happens most of the time, where the electron goes from PS2, down the electron transport to PS1, up and over to NADP reductase to make NADPH. Occasionally, there is cyclic electron flow, where the electrons just kind of go in a circle from PS1 up and down, the ETC, up, down, They never make that right-hand turn and make NADPH. What they do is they go down the ETC again and again, and every time they go down the ETC, we're pushing more hydrogen ions into the pillow of the thylakoid, and we know that the hydrogen ions, when they flow out, are going to create ATP. So cyclic electron flow does not make NADPH or oxygen, but it does make more ATP. All right, I think, and then here's your Calvin cycle. You need to know fixation. What are we missing here? We're missing RUBP, we're missing Rubisco. Reduction is where we're actually using NADPH and our ATP. And then regeneration, we use a little ATP here, but we're not really even worried about that. You also need to know the product of the Calvin cycle. The very official product is a 3-carbon sugar called glyceraldehyde 3-phosphate, or G3P, and that we need 2 of those to make 1 glucose. All right, I think this is your official review. As ever, please let me know if you have any questions. I'm going to be posting a video that goes along with the Calvin cycle that is fantastic. It kind of looks like Dr. Seussy, but it really helps with the math part, and it really makes a lot of sense because it doesn't over discuss all of the intermediates, and it really is a great big picture of fixation, reduction, and regeneration of the carbon acceptor. So let me know if you have any questions, and I hope you have a great day.